Today, we're going to welcome a couple of familiar figures from our community here. Dr. Emily Forbes, a fellowship-trained movement disorders neurologist at the University of Colorado Anschutz Medical Campus, and Dr. Megan Salinas, medical director of the Rocky Mountain Movement Disorder Center. Dr. Forbes and Dr. Salinas are also the directors of the HDSA Centers of Excellence at their respective organizations. And we will arguably and potentially very biasedly, most excitingly today, be speaking a little bit about HD research and promising new therapies. So to get us started, please welcome Dr. Forbes.
Perfect. Can you hear me? Okay, I think it's working. So I am really honored to be able to talk to you today about breakthroughs in Huntington's disease and HD research. An outline of our talk today is I'm going to do a quick genetics refresher. Then I'm going to talk about the positive results from the AMT-130 study. So I think a lot of you have probably heard about this or seen this. So we're going to go over all that data. And then we're going to talk about a few other potential therapies and research, including antisense oligonucleotides and what's recently been called Votoplam, which is an oral treatment that's about to enter a phase 3 study. So, quick refresher, Genetics 101. We all have a complete set of all of our DNA in each of our cells. And our DNA essentially is a blueprint for making the proteins that the cell needs to function. As you might imagine, even though we all have a complete copy in all of our cells, our liver cells are using different proteins than our heart cells and different proteins from our brain cells. And the way it works is all of this DNA is wound up tightly in the nucleus of our cell. And when our cell needs a gene, it calls up that DNA. So you can see that on the left. So it calls up that DNA, it gets a copy of the gene that it needs, and it turns it into a messenger. And that messenger is RNA. That's the strand in the middle. The RNA goes out to where all the machinery that turns this into a protein is, so it goes out into the cytoplasm, and that message becomes a protein. And the goal of many of these therapies is to prevent this step from happening. We don't want this message turned into a protein. All right, so the genetics of HD, I know a lot of you are familiar with this, but we're just going to go over it briefly. So this is a CAG repeat expansion. And so individuals who have less than 35 repeats, that DNA message goes on to make a normal-sized Huntington protein. And then that protein goes on to function in a normal, healthy neuron. Individuals who have too many of these CAG repeats, so if it gets above 35, that goes to make an elongated protein. And so this mutated Huntington protein or elongated protein is toxic to our cells. It wreaks havoc in our nerve cells, in our neurons, and it essentially, that long repeat in there, it makes it kind of a sticky protein, and then they all stick together and they aggregate in the cell. And it stops the neuron from functioning normally. And when the neuron goes through that, it recognizes that it's not functioning normally and it gives itself a signal to die off. And if this was one individual neuron, it would not be a problem. The problem is this is happening on a mass scale in our brains because this protein is required in so many of their brain cells. And so if we look at the brain and we take a cut, and so this image is if you take a cut through the brain like this and look at it straight on, there's an area called the striatum. So this area is affected first and foremost in HD. And so on the image on the right, you can see the normal and mutant protein, and on the bottom, that aggregation of toxic fragments of mutant protein that leads to cellular death. And this happens really pretty aggressively in that striatum, and that's a pretty deep part of the brain. So we're going to go back and just look at where we can target that prevents that protein from being made. So this is a little bit more of a busy picture, but it essentially shows you something similar to a couple of slides ago where we're in the nucleus of a cell and we have that green strand, which is our DNA. That DNA gets turned into that messenger RNA. There are some therapies out there and some research out there that you can see in those pink squares, CRISPR and ZFP, which stands for zinc finger protein. Those are preclinical, meaning they're looking at them in the lab and they look very promising. We're not going to really get into those today, but there is work being done on the DNA itself. We are looking more at this messenger RNA. And so this messenger RNA with the abnormal expanding CAG repeat gets made. It goes out into the cytosol and it gets turned into a protein. And this is really what we want to prevent. And we're going to talk about 3 different strategies today. The first, we're going to talk about how we can use what's called an AAV, or a viral vector, to prevent that protein from being made. Then we're going to talk about something called ASOs, or antisense oligonucleotides. And then we're going to talk about small molecules. Okay, so let's move forward and talk about the positive results of gene therapy in HD. So I woke up on September 24th, as I'm sure everyone here did, to this headline that uniQure has announced positive top-line results from a pivotal phase 1/2 study of AMT-130 in people with Huntington's disease. And this is one of the many headlines that was sent to me and that came up on my phone. Before we start looking at these results, which are really extraordinary and we're really excited about, I want to remind everyone, this is an investigational medicinal product. It is not FDA approved as of today, October 18th, 2025. This is not approved by the FDA. It is not approved in Europe. It is positive results from a clinical research study. So I also want to mention this phase 1/2. So what does this mean? And why was it so important to add in the announcement? So there are different phases of clinical trials. We have the preclinical and discovery times where this is essentially in a lab. We're looking at human cells or mouse models or non-human primates to see if there's any sort of compound that can help treat a disease. And if something looks promising, it then goes into the phase 1 study. And phase 1 is all about safety. We give it to a few people, make sure that it is safe and well tolerated. If that looks good, then it can move on to a phase 2 trial, which is when more people are exposed. And again, safety is paramount. It's the first thing that we're looking for, but also any hint of efficacy, or does it work? And if that looks good, it typically goes on to a phase 3 clinical trial, which is when we give it to many people. This is in the hundreds, or in some cases, even thousands of people can receive a study drug. And if that looks safe and it looks like it works, that is typically when a company will ask the FDA to review that data and approve a medication. This is a phase 1/2 trial, so it is looking at earlier data than a typical clinical pipeline because there's such a desperate need in Huntington's disease, and the company feels like their data looks really good. Okay, gene therapy. So we're going to talk a little bit about this. This is a technique that uses genetic material to treat or prevent a disease. So AMT-130. So this is a one-time administration of an investigational medicinal product that potentially has very long-term results. It is delivered directly into the deep brain. And so you can see that on the image on the right. So this is well within the brain, that picture of the striatum that I showed you before, that's a deep part. So this is where this is going. And it has 3 sites on one side and then 3 sites on the other side. The treatment suppresses both the normal gene expression and the abnormal gene expression, because remember, in someone with HD, they have one elongated CAG repeat, and that goes to make a prolonged protein, but they still have one that's normal, typically. The goals of this study are to slow the progression of Huntington's and improve quality of life. So who was in this study? So you can see there are 2 groups of people. In the orange, there's the high-dose group, and in the purple is the low-dose group. We're mainly talking about the high-dose group results today. And so you can see there's a timeline on top, 12 months, 24 months, 36 months. This is the course of the study. So this is how long that they have data for. And you can see high-dose AMT-130 arm, and there's parentheses n equals 17. That means 17 people got this dose. Now, right below it in the gray box, you can see Enroll HD matched external control arm and then N940. What that means is they did not have an internal placebo control in the study. People who got this treatment were compared to individuals from the Enroll HD cohort, and they tried to pair them so they looked similar to the individuals who enrolled. And so this is important to note because most studies do have a placebo control, especially when trying a new investigational product. As you can imagine, It would be very hard to convince people to go through a prolonged neurosurgery with holes drilled into their brain or into their skull and a cannula advanced in their brain with an infusion if there's a chance they could get the placebo. And so there was no placebo control. I will mention, in case anyone has gone through some of this, which has been advertised online or posted online, in the United States, because this trial went on in the United States and in Europe, in the United States, there were 10 people who did get a sham surgery early on, but they were only allowed to basically not know that they got the sham surgery for 1 year. And after 1 year, they were told whether they got the sham surgery or not, because they thought, essentially the FDA said it's unethical to keep people in a blinded group like this for a clinical trial like this. So there were 10 very brave people who underwent a sham surgery. All right, so all the way on the right, you can see N equals 12 patients with 36 months of follow-up as of June 30th, 2025. That means of the 17 people who got the AMT-130, the high dose, 12 people had data or finished at the 36-month mark. So they got up to 36 months, and they analyzed those people. When we have clinical trials, especially ones that are involved in this, it can be very difficult to recruit and have people enroll. And so not everyone enrolls at the same time. It could take years to get enough people for a trial like this. And then right under it, you see the propensity score matched AMT-130 high dose arm. Propensity score matching is a statistical technique that allows us to compare two groups if one is from the enroll cohort and one is from a cohort that is getting the therapy. And so what it essentially means is we want to compare similar people. We don't want to compare someone who's like a young marathon runner to somebody who's older and has heart disease and hypertension and high cholesterol and all these other things. That's not really a fair match. We want to compare apples to apples. And so they use this method to ensure that they're getting people who are well matched. And so this just gives you an idea of ways that they matched. And so the top three are pretty clear. So sex, age, and CAG repeats look similar in the group that got the AMT-130 and the historical controls. And then they looked at a whole slew of other measures that they wanted to make sure that people were similar on going in. Okay. Then they looked at how do we measure results, essentially. So in clinical trials, we really like to have a single outcome that captures disease across multiple domains. So as we know, Huntington's is not just one thing. We have motor symptoms, we have cognitive symptoms, we have changes in function. And so instead of looking at each of those individually, we like to have one composite score that captures all of these. So for that was the composite Unified Huntington's Disease Rating Scale. So this is the first thing they looked at. And this is a composite of the total functional capacity. On the left, you can see this is impairments in occupation, finance, domestic chores, daily living, and care level. So that was part of the composite score. They looked at the total motor score, which is impairment in motor function. They looked at symbol digit modalities tests. So this is one of two cognitive tests, essentially a thinking and memory test. And it looks at attention, processing speed, and working memory. They used a second thinking and memory test called the Stroop word reading test, and that measures selective attention capacity and processing speed. So these were all things that went into the composite score. So now I'm going to show you the first piece of data that was released, which is the composite UHDRS. So the company has said that they have seen a 75% decreased progression or reduced progression based on this data point. So what does this mean? Okay, so you can see on this graph, what we know is down is worse. And then on this horizontal line, it's essentially time. So we have a baseline, 1 year, 2 years, and 3 years. The gray line is the historical controls. Those are people from Enroll HD. And you can see that over the course of several years, they progress. They get worse year after year. Then, if we look at the orange line, which are people treated with AMT-130, we see it also goes down, but there's a clear difference here in how much it goes down. And so this difference, here we see individuals in the Enroll HD cohort went down by about 1.5 points. People who had AMT-130 went down by about 0.38. So that is where that 75% slowing is. One thing to note, this does not appear to stop the progression, and it doesn't appear to reverse any damage that has already been done. So that's important to know. But it does seem, based on this data, that with the composite unified Huntington's disease rating scale, it slows the progression. Again, only 12 people, so very small sample size. And these lines, essentially, you can see there's a line around each. These lines are the probability that the true point is somewhere in between here. So this is what we think. It's hard to have an exact point. You'll see these lines are smaller, again, because they have 568 people that they were looking at. So they had a lot more data, and these lines are larger because we only had 12 people. All right. So the next thing I'm going to talk about is the total functional capacity. So this is something that we measure in clinic. It essentially looks at ability to perform normal occupation, ability to do finances normally, ability to do your own activities of daily living, domestic chores, and if you're able to receive care at home, or if you need help at home or need to go to an institution. So this is part of that composite unified Huntington's disease rating scale. But if we look at it on its own, we also see a difference. So remember, down is worse, and then horizontal is over time. And we see, if we look at the historical enroll cohort, that gray line, those people get worse more than people who got AMT 130. And then I know some of you are thinking, doesn't it look like that line goes up a little bit? It does, but remember, there are only 12 people in this data point. And so somewhere between here is probably where that true data point lies. And we see that there's a difference between where those two lines end. So it looks like there's a slowing, but we can't say much about, is it actually getting better, or is it just within the margin of error here? And so when they say that AMT 130 reduced the progression of HD by 60% based on the TFC, that's what they mean. It's this difference. Okay. The next thing they looked at was that first cognitive test, the symbol digit modalities test, which looked at attention, processing speed, and working memory. So this one, I don't have a by year time point for you. We just have this bar graph, but we can see, again, down is worse. Historical controls got worse more than individuals who got the AMT 130 high dose. They say this is reduced. So this reduction by 88%, this is what that means. That's the difference here. They also say, they also say at 36 months, and you might see this p.057. So a p-value is basically our confidence that something is a true result. We want to make sure this is real and not just by chance. And so we want those p-values to be very low. The lower it is, the less likelihood it is by chance. They tend to be higher, again, in smaller groups. So if we only look at 12 people, this might be a larger p-value, so more likely that it's by chance. So essentially, it needs to look at more people and see how this pans out. And the second cognitive test that they did was the Shoup word reading test, which looks at attention capacity and processing speed. And again here, individuals in the gray were the Enroll HD cohorts. So these people get worse over the course of 3 years. Individuals in orange were people who got AMT 130. Again, I know what you're thinking. It looks like it's actually going up. Again, it's hard to tell from this data, but certainly these are exciting results. All right, and then the last one, the motor score. So then they looked at motor score, and they kind of went through it in the way that I presented it, where they looked at the composite unified Huntington's, and if that was positive, they looked at the next thing and the next thing. I think we all would agree that if Huntington's disease was solely a disease of motor change and no other symptoms, it would be a whole different thing. But the motor symptoms are really important to treat, and they can be very quality of life limiting for people. They see that, we switched it up a little bit for you. In this slide, higher up is actually worsening. So if it looks like everything's going up, it is because that's what's worsening in the way this data is displayed. So people in gray were part of the Enroll, the historical controls. They got worse, and individuals who got the AMT 130 got worse. This was not statistically significant. So we see that p-value down there. So there's kind of a higher chance that this data fell like this just by chance. Essentially, the way to get more data on this is just look at more people and see how it pans out. Okay, now I'm going to mention something that we don't talk about that much in clinic, but is certainly used in research a lot. So this is the natural history data that shows NFL. So NFL here, it's not the Broncos, it's neurofilament light chain. So this is a marker of neuronal stress. So we know even in individuals who are healthy, this goes up as we get older. It goes up a little bit. In Huntington's disease, this goes up a lot. So it goes up more in Huntington's disease. I want to mention this is not specific to Huntington's. This is a general marker of stress on our brains. People who have Alzheimer's will have elevated levels of NFL. People who have MS, or even people who've had brain surgery for other reasons, their neurons will put out this distress marker. So this increases in HD by about 10 to 15% per year. So we see that it gets worse in HD, and it's a marker of stress in our brains. In people who got AMT 130, they actually had reductions in this neurofilament light chain at 36 months. So this slide's a little different because what I'm showing you here is those people who got the high dose AMT 130. This is what we've been looking at the whole time. Also people who got low dose. We haven't really talked a lot about the low dose. The data is more interesting for the high dose, but they did put this out just so people can see. The control here is the baseline of these individuals. So they looked at them before they got the AMT 130 and then at 3 years. And we see that in both groups, this decreased. So that is very promising. All right, and then they have a whole large matrix and chart of adverse events or adverse effects. So whenever somebody's in a clinical trial, we keep a very close eye on any adverse effects that might happen. And so what we see here is headache, procedural headache, procedural pain, post-lumbar puncture syndrome, procedural complication. A lot of these adverse effects have to do with the actual procedure of getting the therapy or the lumbar punctures that are required as a part of clinical research. People also reported anxiety, constipation, insomnia, back pain. I don't know if anyone here has participated in a clinical trial. If you have, big thanks to you. I think you know that when you're in a clinical trial, we like to know everything that's going on. If you were visiting family and your cousins had a cold and then you had a cold, we want to know about that cold. If you twisted your ankle, we want to know about that. Everything gets recorded. And so this is a part of that. All right. So went through the good news. Now comes the a little bit more difficult news is how is AMT 130 delivered? So I mentioned this before. This is a gene therapy delivered directly into the striatum with the goal of slowing disease progression by interfering with the production of that toxic Huntington protein. It is delivered once via neurosurgical procedure. I think there were some things here. Okay. What are they actually delivering? So there are two components of this treatment. So the first is a viral vector. It's an adeno-associated virus. So essentially, we live in a world where there are millions of viruses out there. Some of them affect us, many of them do not. So AAV is one that's thought that does not affect humans. It does not cause problems in people. Viruses are able to infect us because they use these spikes on their outer shell to trick our cells into letting them in. And then an active virus will then go in and hijack our cell's own machinery to make more viruses and essentially just produce more viruses. And that's why we get sick from viruses that do affect us. But scientists have developed a way to essentially program a harmless virus, like this one, to use our cell's machinery to make a therapeutic medication. And so essentially what they do is they take this virus and they keep that shell so that it can get into the cell. But instead of having viral particles in there, they take those viral machinery and viral instructions out. And instead, they put in a piece of genetic material that they want. So this is a microRNA. It's a small piece of genetic material that can prevent the production of an abnormal Huntington protein. So they put those instructions in a virus. They send it in. It is able to get into the neuron or the brain cell and then go on and produce this helpful treatment. Once the neuron has been treated, it can, in theory, continuously make its own Huntington-lowering molecule, and this should result in reduced production of the Huntington protein. The goal here is a very long duration, potentially even for life. We only have data on 3 years because the study has only gone out to 36 months. So we don't actually know how long the treatment duration is, but it potentially could be very long. Okay, so some immediate questions that I'm sure a lot of people here have, that I have. Will this be approved? So this is going up for FDA approval in the first quarter of 2026. And so we're just going to have to wait and see what happens with that. It's possible that the FDA will say, we'll approve it, but for only some people. They have to meet certain conditions or be in a certain phase of disease. It's possible they have some other comments on it. It's possible they ask the company to do something else or more data. So we'll just have to wait and see. So the next is who will be eligible for this treatment. We'll know more about that if the FDA approves it. We'll get that guidance. The question is, should we treat people who are pre-symptomatic and have no symptoms that they can detect? We don't know. We'll just have to wait. I wish I could answer that for you today because I'm sure that's a very pressing question for everyone here. Where will this treatment be delivered? So as you can imagine, this is not something you can go into like your PCP or local neurologist and pick up. This is pretty involved. So we think this is going to be done at big centers that already have neurosurgery capacity and the ability to deliver this. And so there will probably be specific centers around the country. Here, we are already talking with our neurosurgeons about a path forward for this in case it does get approved. And I'm sure many HD centers around the country are doing the same. And then, will this be covered by insurance? So a treatment like this will likely be cost prohibitive for almost anyone if insurance doesn't pay for it. And so we'll see what the FDA says, but once that announcement is made, we'll know more about the insurance coverage. Okay, so let's talk about next gene therapy, ASOs. And so remember, this is kind of our scheme. We have our DNA all the way on the left in green. It gets turned into a messenger and sent out to become a protein. We want to prevent that protein from being made. And so we're going to focus on ASOs. What is this? This is an antisense oligonucleotide. What does that mean? Well, our DNA in the nucleus over there on the left is pretty safe. It's blocked off from the rest of the cell, and it can just sit there for our entire lives. And when a piece or when a protein is needed, so say our neurons, our brain cells need a specific protein, it will call on that DNA to find the specific instructions for the protein, and it will make a copy of it and send it out. So that's the mRNA. So once that messenger RNA, the blue line, gets out into the area of the cell where there's the machinery to make a protein, it's pretty vulnerable. It is a good place to target it. And in fact, there's something called an ASO, antisense oligonucleotide. That is a little piece of nucleic acid that is complementary and binds. And when this binds, your cell knows that this is not normal. And its response is to destroy it before it ever becomes a protein. And so that's how it prevents certain types of messenger RNAs from becoming proteins. This strategy has worked successfully in other neurologic conditions, and they're looking at it in HD. Now, after the first research study, you might wonder, how does this one get into the brain? So this one's a little bit more accessible. So this is like a lumbar puncture, if anyone's had a lumbar puncture, where they go into the lower back and they put a needle and they advance it into the area where there's cerebrospinal fluid. That's the fluid that coats our brain and our spinal cord, and that's within the central nervous system. So unlike a lumbar punctures pull fluid out. This would push in a therapy, and that's how this accesses the brain. So this is the Generation HD2 study of tominersen. This was a phase 2 study of placebo, or this is a phase 2 study of placebo versus 60 milligram versus 100 milligram dose of this ASO. Individuals who are enrolled are 25 to 50, and with prodromal or early HD, meaning they have HD that are just the very first hint or first signs of symptoms. The goals of this study are to understand the side effects of this medication and its administration, look at changes in the amount of mutant Huntington protein in spinal fluid, any changes in MRI brain scans, and changes in function. So, for example, ability to think, move, perform daily activities. And so there were updates in April. So April 2025, the company released some updates They had an independent data monitoring committee. So whenever these clinical trials go on, they have just a separate group, an independent group that's not really involved in the company. They look at safety data, and they found no safety concerns. So that was great. The trial now focuses on the 100 milligram dose. So they suggested everyone who was getting it just get the 100 milligram dose. This was because of interim data that suggested a potential benefit for Huntington's disease. And so that's great. This trial is expected to wrap up in the next year, and then hopefully soon after that, we'll get some data from it. So this is good news too. All right, last gene therapy we're going to talk about, we're going to focus on small molecules and something called splicing. So let's talk about that. So this means that essentially we can provide a drug that alters the instructions to make a protein by changing the code. And so we see this is the normal code. And what we can do is we can insert an extra piece of nucleic acid, so of material, into this messenger as it's being processed. And this additional piece of information essentially acts as a stop sign, and it tells the cell to just get rid of the whole thing. And so this is how it prevents the protein from being made. So this trial recently gave a name to this therapeutic. It's called Votoplam. This is an oral medication, so a little bit easier to access. So oral medication, the phase 2 trial of this was recently completed, and our plans for a phase 3 study. So that's exciting. Let's look at some data from the phase 2. This is 12-month data. And so what we see is we're looking at the amount of mutant Huntington protein in the cerebral spinal fluid. Placebo, there was some decrease. The 5 milligram dose, there was a 21% decrease. And in the higher dose, 10 milligrams, there was a 43% decrease. So this is a little bit of older data from June 2024 that looked at this at 12 months. So that was promising. The 24-month data is what was more recently released. So this is exciting because we see a change in that composite unified Huntington's disease rating scale. So in gray, we see, and remember, down is worse, historical controls get worse by 1.1. Individuals on the low dose still get worse, but by a less amount, and then the same thing with the high dose. Now I'm showing you the total functional capacity. So again, down is worse. Natural history in gray, so not an internal, but an external cohort that they looked at. And this got worse by 0.74, and individuals with both the low and high dose still got worse, but by a less amount. And this is after 2 years. Symbol digit modalities. This is one of the thinking and memory tests. And we see kind of the same thing. The individuals who are historical controls got worse, and so did individuals on the low dose, but less so. And there's some suggestion that these guys did pretty well when they were on the high dose. Okay, so now we're looking at neurofilament light chain. And so remember, this is that distress signal that our brains send out. And this increases about 12% per year just based on natural history data, based on what we know of HD. Individuals who got the low dose got worse by about there was a lowering. I misspoke. I'm going to go back. So there was a lowering of data. So here I changed it up on you. Lower is better. The people who got the low dose, there was a decrease in the amount of this distress signal. Individuals who got the high dose, there was even further decrease. And so that is really promising. And there's no gray bar here. There's no control because the control is comparing someone to their baseline. All right, so this was well tolerated, no dose limiting toxicities, good adverse event profile. They are starting a phase 3 clinical trial that will probably start to enroll patients in the spring. They are looking for people who are at the very earliest stages of HD. So they're still functioning at their normal job, doing normal finances, all of that, but have like some hints of motor changes or possible thinking and memory changes. So that's very exciting. So I wanted to thank everybody for your attention. I wanted to thank everybody who's been in a clinical trial and participated, everybody who's driven someone to a clinical trial. It is such a selfless thing, and it is the thing that will provide us with a treatment eventually. So thank you so much for your attention. If you're interested in hearing more about research, any of these trials, or participating research, this is our QR code for our research website. You're also welcome to call our research line, or if you have clinical questions, this is our clinic information. Everyone is welcome to just touch base with me. If you have any questions for me, I will be around for the rest of the morning. Thank you so much.